These students, we will discuss the sample holders or cuvettes or cells used in UV visible spectrophotometer. First of all, we need to understand their objective. The sample cells are used to contain sample, that is our analyte, which is normally in solution form or liquid form, and these red particles, they represent the sample. So radiations are coming from the source and being absorbed by the sample, and some of these which are not absorbed are transmitted towards the detection system, that is detector. These sample holders must have a typical property that they must not absorb any radiations coming from the source which is being studied. For example, if we are studying, say, visible spectroscopy, and we have wavelength range from 400 to 800 nanometer, so our sample cell must be made up of those materials which are transparent in this zone of radiation. They must not absorb any of these wavelengths, because if they will absorb any of these wavelengths, there might be an interference that the sample and the cell both are absorbing the same wavelengths, which will not give us accurate results. So that's the main criteria or main condition of a sample holder. They are very important. There are certain parameters which can be discussed in terms of sample holders, that the variations which are possible in these sample holders are the path length. Normally standard path length is 10 millimeters. Most of the sample cells are having this standard path length, but variation in path length is also possible, and there are certain cells which are available with path lengths lesser than that and even more than that if required in certain analysis. The material by which these cells are made is also very important. For example, they are made up of quartz, glass, and certain plastic polymers. And quartz is transparent in UV region, in visible region, and to the extent in IR region as well, means that it will not absorb any radiations coming from the source in ultraviolet and visible region. Similarly, a glass is transparent in visible region, but it may absorb radiations in the ultraviolet region. So we can say we can use the quartz in the ultraviolet region, and we cannot use glass in the ultraviolet region where it will absorb the radiations and will create an interference. Certain polymers, they are used as material to make these sample cells. Mostly they are used in the visible region, and these sample holders or covers are normally disposable, cannot be used many times. As far as the size is concerned, different size of cells they are available, and it depends upon instrument to instrument. There are some standard size as well, but not confined to a single size, as we can say smaller or bigger size cells are also being used. How much volume a container can take in terms of analysis is also a parameter on the basis of which various sample cells are available in the market. Macro sample cells are able to contain like 7 milliliters to 35 milliliters of sample, and the standard cells which are used, they can take up to like 3.5 milliliter of sample, and there are some semi-micro and micro cuvettes also available which can have lesser than this volume, even up to microliters. Another parameter or property of these cells which is important is whether they are having two transparent sides or their all four sides are transparent. Those which have only two transparent sides are mostly used for the purpose of absorption spectroscopy, and those having all the four transparent sides are mostly used for fluorescence studies. They can also be used for the absorption studies, but they are suitable and required for fluorescence studies as well. Now, as far as various parameters are concerned, we may look into different sample cells here. These are standard sample cells. It is worth mentioning that the sample holders also can have the rubber caps, and the shape can be variable, as can be seen in this picture. These all cells, they have all the four sides which are transparent. And here is another diagram we can see illustrates the geometry of these cells and their dimensions. For example, this very first cell is 10 mm by 10 mm in size. The second cell is of sample capacity where the path length is 10 millimeter but the width is 2 millimeter. Again, it is 2 by 10 millimeter in size where the sample will be kept, and this one has optical path of 10 millimeter and the width is 2 millimeters, and this one is 2 by 2 millimeter sample cuvette. All these five which can be seen here have same external size, that is 10 mm by 10 mm, but the optical path and the container where the sample will be filled is variable, which is 10 by 10 in the first one, 2 by 10 and 2 by 10 and 2 by 10 in the second, third and fourth, and in fifth one it is 2 by 2 millimeter in size. Similarly, we have another set of sample holders with variable shape, as can be seen, and these are quite thin. The path length is 10 millimeter but the width is maybe quite less. For example, these two are variable in a sense that it has 2 clear walls and it has 4 clear walls. The concept of 2 clear walls and 4 clear walls is also applicable to the fact that you can have variable path length if you have a cell with different dimensions. For example, if you have a cell of dimension like 2 millimeter by 10 millimeter, that is 2 by 10, one side is 2 mm, second side is 10 mm, and all the four sides are transparent, then that can be used in both ways. In those spectrophotometers where the path length is 2 mm, one side of that will help, and if there is another spectrophotometer where we have a path length of 10 mm, then this very cuvette can be placed in that very spectrophotometer as well. There are some macro cuvettes which can have volume more than normal standard cuvettes, and as we have already discussed that these macro cuvettes can have up to 35 ml of volume, and they are available in both formations like 2 side transparent and all the 4 sides transparent categories. Again, we have these cells which have variable transparent sides with the caps available, and then this is how the path length can be seen or measured. That this is the area where the light will interact, and it can be seen here, and here in this very cell the path length is 50 millimeters. So these are some unique type of you can say sample holders which will be used for certain specific kind of analysis. And then here we have cells which have different dimensions but are transparent to all 4 sides are transparent, and these can be used in spectrophotometers, as we have already discussed that you can use this sample holder where the path length is 5 mm required. You can use this sample holder where the standard path length, that is 10 millimeter, is required. Similarly, these can be used where the standard path length is 10 millimeter, or there are some exceptional instruments where the path length is 2 millimeter. They can also be used. So we have seen the variety of various cells which are normally used in spectrophotometers and their dimension.